Good afternoon. First of all, I wanted to thank the organizers for giving me the opportunity to participate in this webinar. And now let me introduce myself briefly. I'm Josep. I come from Mallorca, and there I have the opportunity to develop my thesis at two different institutions. One of them was the University of the Balearic Islands, and the other one was the Institut d'Investigació Sanitària de les Illes Balears, which is a research institute. Part of the results and the ideas I will expose today come from my thesis, which was defended last year. The talk I'm going to do today is entitled Disentangling the Genomic Architecture of Psychosis in Families with High Prevalence through System Genomic Approaches. And why psychosis? Well, we focused on psychosis trying to avoid the heterogeneity imposed by classical clinical classifications. As most of you know, psychosis is a domain of symptoms which is shared among schizophrenia, bipolar disorder, and schizoaffective disorder. All these disorders were present in the families we analyzed, and all of them are complex genetic disorders. This means they are the result of environmental and genetic factors. In fact, we know that schizophrenia and bipolar disorder have the highest heritability estimates among psychiatric disorders. What we still don't specifically know is how this genetic component is defined or how the genomic architecture of psychosis is defined. And there have been traditionally two different hypotheses. On the one hand, the rare variant common disease hypothesis, and on the other, the common variant common disease hypothesis. The rare variant common disease hypothesis proposes that the phenotype is the result of a few rare variants of large effects, while the common variant common disease hypothesis proposes that the phenotype is the result of the cumulative effect of a lot of common variants of small effects. Assuming one or the other hypothesis have consequences. For example, assuming an extreme version of the rare variant common disease hypothesis leads us to accept that there is no single psychosis but psychosis. In this sense, it may be a psychosis specific of a single family or even specific of a single individual. On the other hand, assuming an extreme version of the common variant common disease hypothesis leads us to accept that the psychosis is completely homogeneous. Also, the methodological approach is very conditioned by the hypothesis assumed. Traditionally, rare variant common disease hypothesis has relied on linkage and more recently on whole genome and whole exome sequencing, and the common variant common disease hypothesis has relied on GWAS and polygenetic risk scores. In our case, and considering we were analyzing families coming from genetic isolates, we hypothesized that psychosis in our families would be the result of a few rare variants of large effect, always bearing in mind that in these families also common variation is present and common variants also are playing an important role. For example, modulating the effect of the rare variants identified. Analyzing families of high prevalence is a very efficient design, which allows analyzing rare variation without the need of collecting large sample sizes. And also the variants identified in specific genes in these families contribute to the identification of gene candidates, which in the end may help to gain insight into the genetic etiology of these disorders. This is very important considering that psychosis, including schizophrenia and bipolar disorder and the rest of mental disorders, are the fifth cause of disability worldwide and one of the main causes of death. Both suppose a chronic condition which with a profound impact, not only on the person suffering from them, but also on the closest relatives. No wonder that the Psychiatric Genomic Consortium proposed six objectives for the next year's investigation regarding the genetics of psychiatric disorders. 3 devoted to common variants and 3 devoted to rare variants. And the work presented today here, it's related with the 6th objective of the Psychiatric Genomic Consortium, which is systematically and comprehensively analyze around 100 large pedigrees. And this was in fact our first aim, which was to identify multigenerational families with a high prevalence of psychosis coming from genetically isolated regions or psychosis hotspots. And we were able to identify 2 families. The family 1 was identified in 2 villages on the north part of Mallorca, and family 2 was also identified in Mallorca, but this family came originally from the southern part of Spain, specifically from Las Alpujarras, which is a known hotspot for schizophrenia. The second objective with these families was to identify potential target genes affected with rare structural or SNP variants in affected subjects of these families. And for doing so, we carried a SNP array and whole genome and whole exome sequencing of some of the individuals of these families. Using this data, we also wanted to carry linkage analysis in order to provide a statistical criterion to assess the significance of the rare variants identified. And we, as we were mentioning before, also wanted to know the contribution of common variants previously associated to the phenotype in the population in our families. And in order to do so, we calculated PRS scores. And finally, we also wanted to validate the potential variants identified using molecular biology techniques. And now we are going to define the genomic architecture for the families we analyzed. In family 1, we were able to identify 2 different genomic architectures. One for the nuclear family comprising subject schizophrenia 3, 7, 8, and 9, and another one for the nuclear family comprising subject bipolar 12. In the first nuclear family, we were able to identify a rare missense variant affecting the MACF1 gene, which is a very interesting gene which plays an important role modulating the cytoskeleton, and which plays a role in neural cell proliferation and differentiation, neuronal migration, neurite development, and axon guidance. Interestingly, there have been previous reports of rare variants affecting this gene in older psychotic patients. But this was not the only variant identified in family members affected by psychosis in this nuclear family. We were also able to identify 2 duplications affecting 2 interesting candidate genes. In this case, these duplications were also present in some healthy individuals of the family. MACF1 was only present in the affected members of the nuclear family. But we think that these 2 duplications, which affect very interesting candidate genes, which play important roles in neurodevelopmental processes, and which has been previously associated with other neuropsychiatric disorders, may, in contribution and in collaboration with MACF1 variant, be behind the psychotic phenotype of the affected subjects of this nuclear family. These results were published in Frontiers in Genetics. In the second nuclear family, which was in which there was a subject affected by psychosis, which was DD12, we were able to identify 2 deletions in this subject, one inherited from her father and the one inherited from her mother, which were absent in her healthy brother. Interestingly, again, these 2 deletions were affecting genes which have been implicated in neuropsychiatric disorders and which are relevant to the phenotype. There were all the variants identified in subject PD12, but that were also present in healthy brother that may also be contributing to the phenotype in collaboration with the rest of variants identified. And there were also other interesting variants in candidate genes which were present in some of the affected subjects of the whole family, which probably in collaboration with the rest of variants may be also contributing to the phenotype. Regarding linkage analysis in this family, we were not able to identify a significant linkage region, but the linkage regions with the highest lot scores were enriched for ontologies related to neuronal development, neuronal outgrowth, migration, and neuronal postsynaptic density, which are processes which are relevant to the phenotype. And regarding polygenetic risk scores, we observed that psychotic patients carried, compared to healthy controls, more variants previously related to the phenotype in the population. Regarding family 2, we were able to identify a significant linkage region on chromosome 9. Interestingly, this region has been previously associated with the phenotype in other studies. We wanted to narrow down on this region, and we carried association, and we identified 4 loci, 4 SNPs which were associated with the phenotype, which were very close to 2 very interesting candidates, the BRIM1 gene and the astropeptin-2 gene, which are very conserved in vertebrates. It's still little known about the function of these genes, but there are some research studies that point that these genes may be very important during neurodevelopment and that play an important role in neuronal guidance during neuronal migration and also in neuronal plasticity. We search for rare variants segregating with the phenotype in this whole region, and we were not able to identify a single rare variant shared among all affected members of the family. But still, we were able to identify some rare variants that were shared among some of the individuals of the family that may be contributing to the phenotype in this region. These results were published in scientific reports. But in this family, we were able to identify all the variants contributing to the phenotype. This was the case of probably RGAP19, which was the only variant segregating with the phenotype in all the members of all the affected members of the family. This gene seems to play an important role modulating ROA, which is important for the stabilization of the microtubules and for the sprouting of new neurons. Another interesting variant identified in the family was one affecting the reticulum 4 receptor gene. Interestingly, this same variant has been identified previously in other psychotic patients. In the case of the family, we think that this variant in contribution with the linkage region identified and the other variant at RGAP19 may be contributing to the phenotype, but some of the affected members of the family did not carry this variant that has been previously identified in other psychotic patients. Interestingly, reticulum 4 receptor inhibits neural outgrowth by activating the ROA pathway. There were other variants affecting candidate genes that were present in some of the members of the family that may be contributing to the phenotype also, and the linkage regions were enriched for processes which are also relevant to the phenotype. Regarding polygenic risk scores in family 2, which have more family members, show that affected members, psychotic members of the family, carried more variants previously identified to the phenotype at the population level compared to healthy subjects. And also this was maintained when separating the phenotypes into the different diagnostic categories. In this sense, bipolar subjects and schizophrenic patients were the ones carrying the highest number of variants previously associated with psychosis at the population level. With regard to CMVs, we replicated the results that are already being published in the scientific literature. In this sense, psychotic patients carried more CMVs compared to healthy controls of the family. And this was expected, as I said before, in the literature. Some of the CMVs identified in the family were located in very interesting regions. Two of them, for example, one of them was affecting the 3q29 region, and another one was affecting the 22q11 region. Both regions have variants which are among the highest genetic risk factors for psychosis. But usually in psychotic patients here, we identify deletions, and in our case, in this family, we identify 2 duplications. We don't know exactly how these variants are contributing to the phenotype, but probably they may be also contributing to it in coordination and collaboration with the rest of variants identified in the family. So in conclusion, we have identified the genomic architecture of 2 families with high prevalence of psychosis. In these families, the psychosis phenotype is the result of the complex interplay between common and rare variants. The use of system genomic approaches is a convenient methodology for disentangling the genomic architecture of psychosis in families, allowing the identification of potential variants contributing to the phenotype. And these results confirm the high genetic heterogeneity of psychosis. Well, that's all. Thank you for your attention, and I will be glad of answering your questions during the question time.